Hi, everyone. Thank you for joining us today for the next edition of the public webinar series from the Common Fund Data Ecosystem. I'm just going to give it a minute or so for our participants to join. But in the meantime, please just get yourself comfortable. We are going to take Q&A throughout the session and we will address them at the end. So please feel free to use both the chat and Q&A function if you have any questions. And like I said, we'll just give it one more minute so more people can join, and then we'll go ahead and get started. So thank you so much for joining us. All right. Why don't we go ahead and get started? Thank you again for joining us today. A reminder that the public webinar series is designed to educate attendees on the scientific ecosystem. And every month we'll have a representation from a data coordinating center and new developments from the centers that support them. So today I'm just going to walk through a couple updates and then we'll talk to our featured speakers. But this is just a reminder that if you were unable to attend any of the other previous webinars before here, we have a YouTube channel that you can access by using this link or by scanning the QR code with all of our previous webinars in the various channels. So please feel free to check that out if you're interested. As always, if you have any feedback, questions, or you would like to be added to our listserv, please email us at help@cfdeknowledge.org. Here is the upcoming 2026 webinar schedule. As you can see, we're already halfway through the year, and we are excited today to have the Human Reference Atlas here with us. As you can see, the upcoming months, we have August through November, and we'll be taking a break in December due to the holiday. One of the events that we recently just hosted at the NIH was the Scientific Interoperability Incubator for Pan-NIH Resources, or SIPR. I just wanted to take a moment to thank every single person who participated in our SIPR event. We had an incredible turnout of over 70 participants, and each individual was eager to showcase what scientific interoperability meant to them and their resources. As part of this, we developed three pilot projects based off of our conversations. And the first is geared around gene sets, specifically establishing a standardized gene set registry. The second is knowledge graph integration, so creating and connecting module across resource knowledge graph components. And the third is an MCP gateway for NIH resources, where we'd like to identify and connect MCP servers and or similar services for NIH funded resources. And so as we go on further into this project, we're looking for community members who are interested in participating in these pilot projects. And we're going to be developing working groups and efforts for these three areas. So if you're interested in participating in these pilot projects, or learning a little bit more about SIPR itself, please feel free to email us or contact me directly. We'll be giving more information as this comes a little bit more forward. But for the meantime, just wanted to reiterate to everyone that the opportunity to work on these projects is available. One other event that I just want to highlight is the Anvil Community Conference, which will take place on August 31st and September 1st at the Broad Institute. And so the registration is now open, and this will basically talk about the Anvil resource, which is a secure cloud environment for sharing, computing, and collaborating with genomic data. Just wanted to note that the abstracts are closed, but if you are interested in participating, you are more than welcome to do so. Lastly, just a reminder that the Decoding the Data Ecosystem podcast, hosted by Dr. Alyssa Dillman from the Training Center, is available. And this is more than just a podcast. It's really a community for anyone passionate about the potential omics research to solve complex biological puzzles and address pressing health challenges. Whether you're a seasoned researcher, a student starting out, or you're just curious about the future of biology, this podcast offers insights, inspiring stories, and practical advice to guide your journey through the world of omics. And so, as you can see, there's 18 episodes that are available on wherever you get your podcasts. And so, with that, I'll be handing it over to the Human Reference Atlas, who will be presenting today on all of their recent work. And so, with that, will hand it over to Daniel, who is going to be kicking us off.
Good morning, good afternoon. So Daniel will share the slides, but I will give the introduction. Then Daniel will present the methods and details of the work we are presenting today, and then Andy will follow up on that. So it's a true pleasure to be here. Thanks so much for having us. The Human Reference Atlas now serves many different efforts, and so that's why this is the title, because we couldn't list all 25 different consortia that we are serving. But the talk itself is on endothelial cell environments across 12 organs, a total of 47 million cells. And then we are also working on an antibody panel designer for spatial omics data that might be of interest, and we are looking for collaborators. And so on the next slide, you will see more information on what is the Human Reference Atlas. I think most of you know, but just for those which are new to the CFDE and also to atlas making, here we have an effort that was funded out of HubMap initially, and then also SENNet and KPMP and GTEx and many other efforts that are also very familiar to the CFDE ecosystem, which is multi-scale. So we believe you have to come from the human body down to protein level. Multimodal, because we are registering tissue across these many different spatial dimensions. It's three-dimensional because human function unfolds in 3 dimensions. So we can't just work with 2D tissue if we want to, for instance, use the vasculature as a common coordinate framework that is going across these many different spatial scales. And ultimately, we had to agree on how to name and organize the anatomical structures in the human body. And that wasn't easy. So we have these so-called ASCT plus B tables, which have a tautology of anatomical structures down below in red. And then we have a typology of cell types. And Daniel and Andy will both present on the supertree effort. So I think there will be a lot of interest to figure out how to compare cell type populations, number of cell per cell type plus biomarker expression levels, across different teams, across different organs, across different assay types, and of course also across transcriptomics and proteomics data. So that's a huge challenge we have for atlasing. So we started to work on that together with many of you. And ultimately, you also, of course, need to link all of this to standard terminologies and data structures for describing also demographics, so specimen data, biological structure, and also spatial positions. And by spatial, we mean the size, the position, and also rotation of anatomical structures or cell types. And that all needs to be linked to ontology, so that where knowledge graph come in very handy. And so there are a number of publications, which we have an entire bioarchive site on this, and some of them are under review right now. Probably some of you are reviewers. Thank you for the great feedback we get every single time we submit something. So on the next slide, you see the teams that have been contributing expertise and data and code. Ultimately, we have 50 different software artifacts that got published in a recent Nature Methods paper. And if you create an atlasing ecosystem, then you can actually start to share not just data, but also code across these different efforts. And ultimately, there are 13 other consortia, also some funded in Asia and in Europe, and more than 250 subject matter domain experts, which also come into the monthly working groups surrounding the Human Reference Atlas effort. And we could not have done this without all the support by the Human Cell Atlas, because they connected us to organ experts around the globe. So on the right side, you see some of the consortia that we are working with and the organs they focus on. And many focus on lungs, so that's highlighted here. But there are also some which focus on many, many organs, including GTEx and HubMap and others. And there are some which focus exclusively on the brain, for instance. And that's okay, as long as we can share data and code with each other. On the next slide, there is the recent resources paper. So one core piece we had to accomplish was to agree on terminology. What is an atlas? What is a common coordinate framework? And Atlas is not a well-annotated data set. And Atlas really requires that you have like a latitude-longitude system for the human body so that you can map new data into it, either based on spatial properties or based on biomarker expression value properties, cell by gene matrices, for instance. And so you really need both. You need a reference and a way to map onto this. So on the next slide, you see an overview of how to construct the HIA. Next slide. Yep. So here you have the AST plus B tables on the top. You have then different ways to map 3D scaffolds into it, and we aligned the HRA scaffold with the Allen brain, with the Spark scaffolds, and many other scaffolds that now exist. There's a focus we have on functional tissue units. Here, functional tissue units from the kidney, which are hierarchically nested. And then, of course, there's a lot of machine learning algorithms that can be used to then do spatial segmentation and annotation of transcriptomics and proteomics data. And again, that's where the super tree is shining. And then Andy here on our team, he led the construction of the cell type population work and also knowledge graph, which now is distributed together with the HRA knowledge graph. And Kiki Kong, another postdoc here in our team, she worked on HRA Lit, which brings in the scholarly data, so publications from Medline. So ultimately, you have experts creating data, you have the experimental data which comes in, and then you also have the scholarly publication data. And if they all meet in the middle and agree, then very likely this is a true segment. But in some cases, they don't agree, and then you have to dig deeper and find out why. So on the next slide, you see how to potentially use the data. So one usage is, of course, to register tissue into the atlas so that you can see your data and the set of 11,000 registered data sets. Oftentimes these tissue blocks, which are registered, they're then sectioned into these tissue sections. Then they undergo all kinds of different transcriptomics and proteomics assay types. Oftentimes also H&E. Those H&E images are very valuable for FDU segmentation. Please do collect those. And then, of course, if you have sections, you need to reconstruct that all into 3D. And then you also might like to do cell distance analysis, and that's what Daniel will present on. So this is the number 8 in this figure here. And then ultimately, if you have transcriptomics or proteomics data here in B and C, you can now start to map this into the Human Reference Atlas, and you can even start to predict where that data might come from if you don't have that information any longer. And that's really the purpose of the atlas, to register new data into it, and then also to predict for a registered tissue block what cell type populations are likely going to be in there. And then you can run your assay types and compare with that prediction. So you have a gold standard for successively optimizing the predictions that the atlas can make. On the next slide, you see that registration. So from the 3D registration into a kidney, for instance, predict what cell types via biomarkers matrix might exist in that tissue. Or if you have that cell by gene or cell by protein matrix, you can start to predict out of which corridor it might come, so which spatial location in the kidney. In C, you see work by Gloria Priobo's team on lung, where you have a healthy lung on top and a diseased lung underneath, and you can now use cell distance distributions to understand pathological relevant artifacts. And so you can now compare what specific disease might exist, but also what disease stage might exist. And we have now also worked with skin and other organ teams to see in how far this is more descriptive than some other analysis that can be done easily today. And Daniel will also explain all the violin plots and distance distribution plots you see here. Down under D, there's a really exciting collaboration with John Hickey's team. He has hierarchical cell type distributions and also FTUs ultimately. And so it's super interesting to see that most cells have to work in teams in order to deliver the function they are supposed to provide. And you can then aggregate up these teams into larger and larger teams, including entire organs, organ systems, the entire human body. And so I think the take-home message is that maybe single cells is not sufficient. You need the context in which these cells operate. And you need to also understand the context in which functional tissue units, for instance, and organs operate. So it's a multi-scale connected system. And one of the new projects we're working on, the whole Person Physium project, is trying to understand that function better across these temporal scales, but also spatial scales. On the next slide, you have Daniel's first slide. Okay, over to you, Daniel, please.
All right, thank you, Kathy. Let me go back one slide. So hi, everyone. I'm Daniel. I'm the senior software developer here at CNS, and I have worked a lot across a lot of the HRA to create tools and applications that can be used to further analyze data. So I'm going to first now present here on the endothelial cell environment across organ paper that has been that Kat has written here, and as well as the Cell Distance Explorer tool that we have created here to analyze data. So in this preprint, we analyze 399 spatial omic data sets from 14 studies across 12 tissue types with a combined count of cells of over 47 million cells. So our goal in this paper was to understand how different cell types are organized around endothelial cells. And the preprint for this paper is available on bioRxiv. So here on this slide, you see first a couple of images that were taken directly from our Cell Distance Explorer tool. It visualizes the cell layout as well as the distances between the cell and the endothelial cell. Then in the heat maps here shows you which for which data set, for which tissue we have data sets, sorry. So here is a summary of all the data sets that we used in the paper. We have the data sets cover a wide variety of tissue types, different technologies, and a lot of data sets. So to make it easier to explore all of these data sets that were used from different teams, we created this spatial omics gallery where you as a user can browse the studies, select the data sets, and open them and launch them directly in our Cell Distance Explorer tool, which makes it really easy to find exactly what you're looking for. So now I'm going to talk about the Cell Distance Explorer tool itself. It's easy to use, and if you want to use it with your data, the data format to be able to, for it to ingest your data is very simple. You only need a table with the X, Y, and Z coordinates plus a cell type. So here we see just an example little slide of being used. I'm going to demo the Cell Distance Explorer now. All right. So this is the landing page for the Cell Distance Explorer. I hope everyone can see. So here we first have, you can go to this spatial omics gallery, as I talked briefly about. You can also explore just a couple of examples to just get quickly started and dive right to it. I'm going to show you how to create a custom visualization. So the first thing you start is just click the Use app, and it will ask you to upload your data. So I've downloaded the data already from Snyder's team at Stanford for the large intestine. So I'm just going to upload that. It will, if you have the correct data format, it will detect the data and you don't really have to do anything else. But if you have slightly differences in your data format, it allows you to customize here fully. So you can select what the X coordinate uses, Y coordinates, and so on. Once you've done that, you go to visualize and it will launch the tool for you. So let's start at the left side. So the left side here, you see a list of all the cell types. You can toggle different cell types to only show what you are interested in. You can change the color in order to make it more not transparent, but obvious what you're looking at, like so, and it will update everything. So in the center, of course, is the main visualization. It's something you can drag. It's fully 3D, so you can drag and zoom and rotate it as you like. You have a couple of extra tools here too, such as inspect, which allows you to select a specific node and receive the information for that specific node or cell rather. You also have the select tool, which is very useful if you need to delete something. You can draw a little around it and then hit the, if I can, sometimes it behaves. There we go. And delete the cell like so. Then it also summarizes for you in this violin graph and histogram the properties of the different cells. So in the violin graph, we'll see the distance to the endothelial cell as a violin plot. So we can see that the max distance is 828 micrometers, but most of them are under 30, being the median. The histogram, of course, shows you the number of cells in each distance interval from the endothelial cell. And that's it for the demo of the tool. So let me go back to the slides. There we go. All right. So this tool has been used by multiple collaborators. For example, Jean Fan has used this in her HubMap work for the spleen. Then we have Peter Swordwell's group have used it at Harvard Medical School to explore, for example, colorectal cancer. So you can use this both for healthy and diseased data. This is another example from Peter Sorgels. This is a melanoma of the skin. So one thing we had to solve for this paper is a big challenge when comparing data sets across organs and teams and technologies are that they not always align with the cell ontology ID and they use different annotations at different levels of specificity. So our three-level here hierarchy harmonizes those annotations, so the data sets become comparable. The first level is very broad categories like endothelial, epithelial, immune, and as you go down the levels, they become more and more specific. The harmonization is done through a crosswalk, which is created by human experts to link the different levels. and link them to the proper cell ontology terms and IDs. So because all of these data sets are registered within the Human Atlas, Reference Atlas, using our registration tools and exploration tools, we always know where the tissue originates, and we can compare it across different studies and organs. So here we have just a couple of comparisons that's done. So this is the small and large intestine, and we have compared the distance to the endothelial cells as the way that food, in the same way that food would move through the system, through the intestine. So on the left, you have at the very top of the small intestine, and then it goes down through the small intestine, down to the large intestine and out. We also recently created 40 new registered 40 new anatomical structures for the mouth. And then we performed the same kind of analysis across the oral cavity. And here we can also in this plot see that certain cell types only appear in specific anatomical structures in the mouth. So then we have done the same thing for the lymph nodes. And yeah. And this last example is for a skin data set. And here we can see that most T cells lies within roughly 20 microns of an endothelial cell. So very close to the vasculature. And of course, all of these details can be found in our paper. But we have, of course, worked a lot with Dr. John Hickey at Hickey Labs, and he has contributed to the different papers and analysis. He does a lot of hierarchical cell neighborhood analysis. Okay, so now I would like to switch gears and introduce our antibody panel designer proposal, APD for short. So the goal of this proposal is to simplify antibody selection for spatial omics experiments. So this is an initial UI mockup made in Figma for how just it would work on a at a high level. So the APD would let you choose your organs, your anatomical structures, cell types, and biomarkers. And then it would help you identify what antibodies would be useful to use in your studies for this. We would, alongside, in order to power this tool, create a knowledge graph that would incorporate data from many sources, from CFDE sources, HRA, CENET, and others. It would harmonize the data so that, like our cell type topology, the data would have a consistent format and be usable across different tools. We would then, of course, publish this open access through an API, as well as on the HRA portal and CFDE portal. And we are currently talking with the Adgene and CiteAb to see if we can receive data from them, how they would like this tool to be designed, and what would be best for different stakeholders. And now I'm gonna give it up to Andy to present some upcoming hackathon events.
Thank you very much, Daniel. After having looked at the HRA, our CDE spatial omics paper, and the antibody panel designer, I want to briefly look forward to a series of hackathons we at the IU team are organizing this month. Next slide, please. I put the information for each of these 3 hackathons into the chat. If you are interested in attending any of these, either in person or virtually, please feel free to email me directly. My email address is in the chat and also on the slide. So the 3 hackathons in question are our HRA and whole person physiome. That's the WPP hackathon. This one will actually be next week, Monday through Wednesday. I'll have a few more information bits about this in just a second. Then the whole person physiome team is organizing a jamboree, a virtual jamboree or hackathon on August 24th to 25th. This one will be organized mainly by the Stanford team. And then we are organizing a hackathon in Berlin at the Berlin Institute of Health in Germany, where we are going to be discussing the HRA and its connections to the human phenotype ontology that some of you might have used. Again, the whole person physiome and other European Union research efforts. Next slide, please. One piece that we will be working on during all 3 of these hackathons that's really core and centered to our current work around harmonizing cell types across different organs, tissues, and data sets, and also teams and modalities, is the so-called supertree. This is currently being led by Nicole Vasilevsky, who is working with us through HubMap. And the goal of the supertree is to create a harmonized tree that crosswalks cell types to cell ontology terms. You can see an excerpt of the cell ontology here on the right side. Some of you might have experience with that cell ontology. So the main process for constructing this supertree is to look at the top level cell types in cell ontology that are available. You can see those here on the right side. So there's 32 of those. And the goal then is to map cell types in a tree up to these top level classes. And then we follow the is-A hierarchy in the cell ontology to align each level in the supertree to a node in the cell ontology. So if you go to the next slide, here is what that supertree concretely looks like. It is basically a table where the farther to the right you go, the farther down you go in the cell ontology hierarchy. So for example, in the leftmost column, we see the general term cell, which is basically the root node, which has cell ontology ID all zeros. And then a subcategory of this is the hematopoietic cell with CL ontology ID 988 at the end. And then one of the children of that particular hematopoietic cell is the myeloid cell. And you can see how the CSV structure on the left mirrors the hierarchy on the right. And so we're trying to do this across the entirety of the cell types currently in the HRA in an effort to harmonize across all of these levels. Okay, next slide, please. And this is, of course, ongoing work, and it is also quite complex. You can see we are using our skills as data visualization experts to try to get an understanding of the structure, both the depth and the width of the resulting tree with various network visualization methods that you can see here. This is, of course, also an iterative process in optimizing the supertree, and we are extremely interested in working with many of you here who might be ending up using the supertree in improving it iteratively. So these are the 11 top level nodes we are actually using. You can see those listed here on the left. So this is going to be the top level hierarchy of this supertree. Next slide, please. And then for this very much upcoming hackathon that starts on Monday, these are 3 projects we will work on. And those are just my projects. So we have 20 people in attendance, and each of them is going to bring their own goals for this 3-day hackathon. We rarely get to just sit down and program for 3 days. So I'm extremely excited for these 3 days. So these are concrete ideas and goals that I have with 3 of our confirmed attendees: Deanne Taylor, who many of you probably know, Michael Eden from the IU School of Medicine, and Aran Agmon from UConn Health. The goal here on the left side with this little project is to improve visualizations for knowledge graphs and access. CFDE has been really prolific in ingesting and producing new knowledge graphs. With Michael Eden, we are going to sit down and review cell type populations that Kadi briefly mentioned at the beginning of this talk, specifically looking at the kidney, including with 3D visualization, which you can see visualized in here. Also, by the way, supported by an RO3 from the CFDE or through CFDE. And then on the right side, we want to link mechanistic biomodels from the biomodels.org repository to these 3D reference organs that we have curated as part of the HRA. So extremely exciting stuff, and we are very excited to welcome all of these folks here in person to this hackathon. And again, if you are interested in joining one of these future hackathons, please email me directly. Okay, next slide, please. One more thing. We are going to present basically live results from this hackathon next Wednesday in our Human Reference Atlas working group, which meets once a month. And this time, this meeting happens to coincide with the last day of the hackathon. So if you are curious about hearing live results from our hackathon, please tune in on Wednesday between 11 a.m. and 12 p.m. Eastern. There is a expert registration form on here that Kadi will put into the chat that you can use to register to become a member of this working group. And with that, to the next slide, please. It is, of course, not just the three of us who are building the HRA. It has a whole ecosystem of contributors. And here are just some of the amazing people that we have been fortunate with in collaborating in continuously evolving the HRA. And with that, if you want to go to the last slide. This is the acknowledgement for all of the people who contribute and all of the funders who contribute to the HRA. And with that, to the last slide, please. Thank you very much. We have posted many resources into the chat. Here is also a QR code, and please feel to reach out to us. And with that, we are open to questions. Thank you very much.
Great. Thank you all so much. As a reminder, if you have any questions, please feel free to use either the chat or Q&A feature. If you do want to ask your question over, using voice, you can raise your hand and I can give you the opportunity to talk as well if you would like. While we wait, I have I think a general question for you all. So you've talked about, the different collaborators that have helped with this endeavor. Are there any other communities that are going to be involved in this effort as you move forward? How else do you see others getting involved with the building of this atlas?
Great question. I can start and then Andy can also add. So one way of expanding the Atlas effort is to set up mirrors around the globe. So we have currently 2 mirrors of the data and code going. And Bruce is on here also. He could put the Berlin one or some other one in there. And that's great because then people can add local data and then share it back with the global community. Also makes for great sustainability because multiple copies keep stuff safe. Another way of doing it is that we have also funding from CFAR, which organizes meetings that welcome international scholars twice a year for a week almost, to come together and to really share their data assets, but of course also a code and expertise. And so this has been really wonderful because ultimately Europe typically funds Europe and US typically funds US. It's very hard to get funding globally right now. Another way of collaborating is that we started to work with different teams that have relevant knowledge graphs to add their knowledge graphs. For instance, there's the Human Phenotype Ontology by Peter Robinson. He is at the Berlin Institutes of Health, but also at the Jackson Lab. And so he and his team and our teams are working on interlinking these 2 knowledge graphs, in which case you can then see which genes exist in which organs and what goes wrong across the developmental axis, right? In some cases, you have a gene that gets expressed at a certain age, let's say 11, and when that gene starts to be expressed, some developmental change happens, and whoever has that heart would actually not do so well and potentially die. But if you start to have an operation with 10, then you don't have that problem. And so it's really important ultimately to look at adult. Yes, that's what we have been focusing on, but also go into the developmental and even embryo stages, because many things are created there. And ultimately, it's good to have the entire life trajectory captured if you want to go over to digital twins and ultimately use that data, use these atlases to really understand how we age healthy, ideally, and if we get sick, how to get back to a more healthy normal. And Andy, Daniel, please do add. You have your own collaborations, Andy.
Yeah, I put something into the chat. We recently published a preprint on our Human Reference Atlas Organ Gallery that many of you might have seen. The Organ Gallery is a virtual reality application where we harmonize data sets from 6 teams across HubMap, ZenNet, and CFAR. The paper was also co-written by 7 early stage investigators, which I'm especially excited about. And as part of this, we created a series of demonstration videos of how we harmonized all of these data sets into the HRA, which is basically like a little nature documentary with data providers narrating over their data as I show them in VR. So this is all in the chat. So it's a small slice of how we try to extend the HRA, but one that we're extremely happy about and proud of.
Great. Thank you so much. Okay. I know there's one other question in the chat, but I just want to follow up on the expansion of it. So you had mentioned knowledge graphs. Are there any other knowledge graphs or knowledge graphs like the data distillery, which is at CFDE? Is that something that you're interested in or could be a future collaboration?
Absolutely. Yeah, I think the CFDE paper currently in writing um, features how these different knowledge graphs interlinked. The HRA knowledge graph is regularly ingested into the UBKG, Jonathan Silverstein, and he is collaborating, of course, with Diane Taylor. Diane just arrived here in preparation of the hackathon next week. And so her petagraph effort is super relevant for what we are doing. And so we are very interested to collaborate with her next week. And she just told, I want a code. So Bruce is on it. I think they are going to have a good time at that hackathon. And as Andy pointed out, oftentimes you don't get so much. You're just a meeting, you're giving talks, and having a dedicated set of 3 days to just code together across teams is super exciting for many of us.
Yeah, that sounds great. There is a question in the Q&A, which asks, in the overview slides, it appeared there were no bone cells included in the atlas, only bone marrow, which is not bone tissue per se. In the super tree slides, there was a mention of a scalogenic category. Does the HRA capture bone cells? For example, there are 42 billion osteocytes that are resident in the skeleton.
So Andy, you would have more information what's an HRA pop. I do know that we are now expanding the HRA to also cover relevant organs and organ systems that we need for the whole person physiome project. I put a link into the chat on that new effort. And here we are looking at 10 raised to the power of 10 spatial dimensions, 10 raised to the power of 10 temporal dimensions, so from fast nervous signals to lifelong bone structure changes, and that across 14 physiological systems. And the musculoskeletal system is absolutely part and parcel of this. You need that. Can't do it without. And so because of that project, we will bring in some more data, but I know we also have femur in the Human Reference Atlas, so you can register into this. We had an entire team working on knee, and there is also quite a bit of data from Vidyani and others in the ZenNet project. So this data is coming in, and you will get to see this in HRA Pop also.
Yeah, and adding to this, I am putting some links into the chat to the 3D reference organs that we currently have pertaining to bones broadly. In order to map these cells into the HRA, we typically need a 3D reference organ that represents the anatomy so that the data sets can be registered in there. I put links to the pelvis and knee in there. These are open source 3D reference organs that we modeled, mostly based on visible human data with expert medical illustrators with the input from subject matter experts. So this is currently the stock of 3D reference organs that we're trying to expand. And thank you for this pointer to these millions of cells we're going to investigate and see how we can map them into the HRA.
And Nancy Rushman is also here on the call. So Nancy, you might like to reach out as well. She has been supporting teams that are interested in registering their data into this Human Reference Atlas.
Any other questions for our speakers?
I believe Andy and Daniel might be coming to the next CFDE meeting, which I just learned is not going to be in DC, but in California, La Jolla, or some San Diego place. Looking forward to this. I always believe that just parallel play is not enough. You have to try to start integrating these data sets, integrate code, which is not easy because some code is written by postdocs working in experimental labs. Others is written by professional programmers. So there's a difference in what you get there. But then ultimately you need quite a few different algorithms to construct these atlases, to use them, and then to also serve very custom applications.
Yeah, and I put into the chat one more time the expert registration for the HRA working group, which is the best way to regularly engage with us for building the HRA. And again, where we will report some updates from our hackathon next week. And yeah, if you are in California for the next CFD meeting in fall, please come and say hi. And would be great to meet you in person.
And Andy is typically bringing his hip VR headset, and then you can see the entire set of organs and cells and FTUs across scales. And that's a nice walk on the wild side.
I can confirm that is a very interesting way to look at things, and I'm just in awe every single time that I see the updates that you have for that. So I concur. All right, I'll give it another minute just to see if there's any other questions. But just a reminder, while we're just waiting for some other questions to come in, that our next session will be August 28th. So just a reminder that, that'll be in the next month. People are just saying thank you for the informative session. So I would agree. Thank you so much for joining us. It looks like there aren't too many other questions, but if anything comes up, please feel free to contact the HRA using their contact information that's on the screen here, or you can contact help@cfdeknowledge.org, and that'll come directly to me, and I can guide you to the contacts. And Andy also just sent in the contacts in the chat as well. So thank you so much for joining us today, and thank you to our featured speakers. We greatly appreciate you taking the time out of your day to present. So thank you, everyone, and have a great rest of your Friday.
Thank you for organizing.
Thank you.
Bye.
Thank you.
Bye.
Bye.